Sri Sai Ram. Welcome to Sri Sathya Sai Loka Seva Gurukulam. Today we are going to see the continuation of the chapter Building Blocks of Life, Cells and Tissues. It is part 5. As usual, we will start with the home assignment answers. Last time it was asked the difference between plant cell and the animal cell, right? So here you can able to see some of the differences were listed. In the plant cell, the cell size is large rectangle, and in plant it is smaller and also it is oval in shape. Cell wall is made up of cellulose, and here the cell wall is absent. And in plant cell we can able to see a single large central vacuole, whereas vacuoles are absent. If it present, it will be very tiny and small, and it is scattered throughout the cytoplasm. The Golgi bodies are diffused in plant cell and it is called as dictyosomes, right? It is well developed in animal cells and it is present near the nucleus. The other differences we can able to see few are here. The centrosomes or centrioles are absent, whereas in animal cell it is present. Plastids are present, whereas in animal cell it is absent. The major thing is the storage granule differences, right? Here the starch and oil are the storage material, food reserve in plant cell, and in animal cell it is glycogen. These are some of the differences between plant cell and also the animal cell. Now we will proceed further. What are we going to learn today? We are going to study cell division, the types of cell division. In elaborately, we are going to see mitosis. First, we will see what is cell division. The cells wear out and need to be replaced. What do you understand from that? The cells are getting damaged and we are falling down or something is happening, right? We are growing and naturally also some of this old cells has to go out. So this wear and tear mechanism is very common in any of the living organism. So whatever cell is damaged or getting old has to get replaced. So that is what the major event which is happening in the cell division. The new cells are required for all the living organism for various purpose. The first one is repair of any cut or injury. We could able to see scar formation, right? When we are kids, we used to fall down and we'll hit somewhere, all those things, right? So we'll have some of the scars. How the scars have formed? The scars have formed because we fall and then we got a cut, we got an injury, and our cells started dividing and replacing the damaged tissue, and it is replacing the damaged tissue with the scar tissue. So we can able to see the cell division which is happening in any of the organism when there is a necessary. The next one is growth. When we are very tiny, we will be very small, right? We will have very less number of cells. As the organism grows, it has to tend to increase its body mass, right? We were like tiny babies when we were born. Now we are all very big. How we are big? How we became very big? Because we started eating all what we have eaten, that is getting converted into our own component, right? We can able to see the morning breakfast is going to become you, right? In some days, right? It is not exactly it is going to do, but slowly it will gradually increase in size. That is, the growth of the organism is possible only because of the cell division, and it is a process of reproduction. How a cell division can be a reproduction? Did you heard of the yeast? The yeast is the tiny organism which is used in the bakery and all, right? It will make the dough little fluffy buns and all they will add, right? So this yeast are a single cell organism. If the cell divides, then it is a process of reproduction, right? Two cells, two yeast we are going to get if one yeast cell is dividing. So that is a process of reproduction in few of the organism. So these are the various things what we are going to see in the requirement for the cell division. So new cells we are getting it because of the process of cell division. Now we will see what are the types of cell division. There are two types of cell division which is present in the plant cell and also in the animal cell. So what are the two different kind of cell division we can able to see? The first one is known as mitosis. What is mitosis? The cell gives rise to two identical daughter cell. One cell has to divide and the exact replica we can able to see, that is known as daughter cells. Okay, in nature only female are the pertains to the growth and also the reproduction in the environment. So it is known as daughter cells. Okay, so these two daughter cells it is forming, and these cells are needed for the growth, repair, worn out. It is present in our body cells, wherever we can able to see face, hands, everywhere we can able to see these kind of cells. When it is undergoing division and growth and repair, then the process of mitosis is happening inside the cell. Because of that only we are getting two cells. The next type of cell division is known as meiosis. In meiosis, the cell divisions involves in production of the gametes or the sex cells, and which gives rise to the gamete in the female and also in the male organism. You can able to see the pollen grains, all those things, right? So that is all because of the cell division which is happening inside the flowers, which is known as meiosis. Okay, there are two types of cell division. Now in this presentation we will see what is mitosis and how it occurs. So to start with, it is a major event that is taking place in the cell division, and which is similar in both plant cell, animal cell, and also yeast also I have mentioned, right? It is a fungal cell. So in any of this organism, if it has to undergo division, this is the process by which it is undergoing division, right? So it is having the similar kind of organization and the event which is taking place in all these cells. Mitosis is described as karyokinesis and cytokinesis. In karyokinesis, we have four phases. In karyokinesis, what is happening? Karyo pertains to the chromosomal part, the DNA part. This DNA has to get separated among the two daughter cells, then only it will become identical, right? So the karyokinesis has to happen first, which is followed by cytokinesis. This is the division of the cytoplasm and other cytoplasmic organelle. So these are the two major events which is taking place in the mitosis. In karyokinesis, we can able to see again four phases: prophase, metaphase, anaphase and telophase. These are the four phases. Okay, this is how the mitosis is described. Now we will see the events which is taking place, right? Eventually we can able to see that when a cell is present, right, we can able to see the cell which is mature and then it is about to divide. So it is ready for the division. That time what are the things happening? The nucleus, we can able to see the nucleolus and also the chromatid which is present, right? We can able to see the dispersed DNA which is present. It is all spreaded out DNA. This DNA is known as chromatin threads. These chromatin threads, where it is present? It is present inside the nucleus. It is needed for the cell protein synthesis, other control, the entire control of the cell is under the nucleus, right? So all those informations are present in this chromatin threads. So we can able to see a dispersed, it is loosely arranged inside the nucleus. And we can able to see the centrosome. Here we are talking about the animal cell. So you can able to see a prominent centrosome which is present adjacent to that of the nucleus, and the cytoplasm is also present. This is a normal cell which is about to undergo division. So what is happening during that time? This chromosome has to separate it among the daughter cells, right? So this chromosome materials inside the nucleus, now it is becoming condensed. Condensed means it is becoming little thickened like structure. It is thickening because of the organization of proteins and also DNA with that. Okay, because of that the condensation which is happening, and that chromatin threads, now it is converted into chromosomes. If you wanted to separate out the threads or anything, okay, you can able to see the hair stalks and all, right? So how they will transport from one place to another after harvesting? They will make a bundle, right? With the bundle, it is easy for us to transport. Similarly, here the chromatin is like a full straw which is present throughout. This straw has to get transferred. So, the nature itself is bundling up. So, this bundled up chromatin threads, now it is known as what? The chromosomes. Okay, that is going to form when the cells are going to undergo division. Now, what are all the other things which is happening? The nuclear membrane which is present around the nucleus, now it has started disappearing. That is, it is disintegrated into tiny parts. It is not completely dissolved, it is disintegrate into tiny parts. So, the chromosomes now we can able to see which is freely floating in the cytoplasm, right? Because the nuclear membrane is disappearing, which is followed by the centrosome. These are the organelle which is present only in the animal cell immediately next to the nucleus, right? This centrosome, what it is doing? It divides. Okay, it is an intact centrosome. When the cell is about to undergo division, we can able to see the centrosome divides and it is becoming two equal halves. The centrosome, it is divided into equal parts which is known as centrioles. Each of the centrioles will migrate away from each other. One will move up, the other one will move down of the nucleus. So, it will migrate towards the opposite side and also it orients the spindle fibers. Spindle fibers are small thread, okay, that is formed from this centrioles. These are going to generate the spindle fibers which is present in the cytoplasm. These are the initial events which is taking place inside the cell. The entire process is known as prophase. And the spindle fibers appears between the centriole. Now we can able to see the spindle fibers have started forming here, right? So now the next phase is known as metaphase. In metaphase, what is happening? Each chromosome consists of two chromatids. You can able to see, right? The two chromatids which is present, two chromatids which is getting attached. These two chromatids are held in the center by a small structure which is known as centromere. Okay, don't get confused centriole with the centromere. Centrioles, now it is present in two opposite poles only in animal cell, whereas the centromere which is present in the center of the chromosomes. Okay, this chromosomes arranged in the center equatorial plane. This middle or the central equatorial plane, when the chromosomes are arranged, this phase is known as metaphase. In the previous, chromosomes have formed, right? Now it is getting arranged. We can able to see the spindle fibers are also going. Can you able to see the lines which is coming? It is originating from the centrioles in the both the opposite poles. Now these spindles will get access to the chromosome and it will get attached to the centromere region. These are the events which is taking place in the metaphase. Now we will see in anaphase what is happening. In anaphase, the centromere splits. Now the small fibers which is getting attached to the centrioles, right? Because of the pulling action from both the direction, the centromere, now it is getting split. The chromatids, now it is known as daughter chromosome, because of the break which is happening in the chromosome, it is becoming a daughter chromosomes now. These daughter chromosomes are now it is having its own centromere, and this chromatids, now it is going to get separated from each other. Okay, the chromosomes which is getting attached to one of the fibers which is coming from the centriole, it will pull it towards it, right. So, in that counterpart we are having the another sister chromatid which is getting separated to the opposite pole. So, we are dividing the entire chromosomes into two equal parts, and it is known as daughter chromosomes, and the daughter chromosomes moves in the opposite direction. You can able to see one is moving towards this, the other one is moving towards the other end. So, we can able to separate the chromosomes towards its opposite side because of the spindle fiber which is getting attached to that. Where the spindle fibers are coming from? It is coming from the centrioles. Okay, this is known as anaphase. Then it is followed by a telophase. Now, in the telophase, you can able to see the chromosomes are almost reached the pole, right. You can able to see, since it is a animal cell, we can able to see a separation also started forming at the end of the telophase for the cytoplasm. In telophase, the chromosome loses its identity again, and what it is going to become? It has to become the chromatid. This chromatids, now it is grouped in the opposite pole. It is not in the middle anywhere. No, now the chromatids are started forming near the centriole region towards the pole of the cells. Now, the nuclear membrane which has disappeared before, right, in prophase, it is reappearing again around the chromatid which started coming and gathered near the pole, and it is forming a nucleus like structure. It is not yet formed, it is about to form. So, that is happening at the end of the telophase, and at the end of the telophase immediately you can able to see the cytoplasm is tend to separate, that is known as cytokinesis, right. In cytokinesis, what is happening? It is the small depression which is forming near the end of the telophase. Now, it deepens. It deepens and deepens. Since the furrow is deepens in both the direction, how we will make roti or chapati? That time we will divide the dough into two equal parts, like that. It is, the cell is having two from the opposite direction, we can able to see the furrow which is forming and which divides the cells into two identical equal daughter cells. Why we are telling identical? Because here the chromosome material, whatever we are considering, exactly what is present here, the same thing is going to present here. In our cell, all the skin cells will have the same kind of genetic material, right. So, we can able to see exactly same how it was present, the size, shape, everything will be same. So, here we can able to see the centriole is also getting separated along with the chromosomes are getting separated, right. When the cytoplasm is pinching off, we could able to see some of the mitochondria, endoplasmic reticulum, Golgi apparatus, ribosomes, everything is also getting splitted out among the two daughter cells equally. This is all the events which is happening in the cytokinesis. So, in the cytokinesis, we can able to see in the middle, we can able to see the furrow which appears in the cell membrane, and the furrow deepens and which divides the cells into two identical daughter cells. Now, we got two identical daughter cell at the end of mitosis. Now, we can see some of the differences are happening between plant cell and animal cell. The entire events are happening same, but there will be a subtle difference in plant cell and animal cell. The plant cells, there is no centrosome present, right. So, the spindle fibers are just forming in the cytoplasm, whereas the centrosomes are present in the animal cells. Upon the completion of mitosis, the cytoplasm has to divide, right. That is, the cytokinesis has to happen, right. It is a furrow which is forming in the animal cell that we have seen. This kind of furrow cannot able to form in the plant cell. Why? In the plant cell, a rigid cell wall which is present around that, right. So, it won't allow the cell to do this furrow formation or cleavage process. So, instead of that, what a plant cell is doing, plant cells will form a cell plate. This cell plate is newly formed. This is exactly similar to the cell wall constituent, and this new cell wall is formed in the center. in the cytoplasm in the middle, where the chromosomes were arranged in metaphase, right? The chromosomes are arranged in the center equatorial plane. In that place, the cell plate will form, and the cell plate from the middle, it will extend in all the 360 direction, and it divides the cells into two equal half. The furrow formation we can able to see only in the animal cell, whereas cell plate formation we can able to see in the plant cell because of the rigidity, and the cell wall has to formed. So, the cell wall is forming in the center as cell plate in plant cell. And now we can able to see at the end we are getting two daughter cell, either it is plant cell or animal cell, we can able to get it. Now we will see what are all the significance of mitosis. The daughter cells receives the same number of chromosomes as that of the parent cell. We can able to see it is identical cell, we are getting it at the end of mitosis. Two cells we are getting from one cell, right? So, they are very identical, the same number of chromosomes are present. That is because it is known as equatorial division. Equally it is dividing. Equatorial division is the other term which is given to mitosis, in which the daughter cells are identical, exactly same, identical, and each other when we are considering its parent cell, you can able to see they are all looking alike. Mitosis helps in wound healing, replacement of any lost cells during the wear and tear mechanism. You can able to see the lizard tail also grows, right? So, you can able to see the replacement, regeneration is also possible in few organisms. So, mitosis helps in wound healing, replacement of cell lost during any wear and tear mechanism. Okay. The next is it is responsible for the growth of the organism. So, we can able to see the organism tend to grow because addition of new cells are taking place, which makes the organism bigger. And it is a method of asexual reproduction in single cell organisms such as amoeba. Amoeba is also one of the animal, right? It is a eukaryotic microorganism, small one celled organism. In that it is a method of reproduction, because one amoeba divides into two amoeba means we will get reproduction, right? Two organisms has produced. So, here it is a kind of asexual reproduction. One cell itself is dividing. There is no male and female present. So, we have to use asexual kind of reproduction which is taking place in the amoeba. These are some of the significance of mitosis. With that, we will have some of the home assignment questions. What are the events in mitosis? Write any one difference between plant cell or animal cell in terms of cell mitosis. Draw and explain metaphase. It can be asked any phase, right? Prophase, metaphase, anaphase, telophase, anything. Draw and explain it is also. You are expected to draw a diagram and you have to explain the events which is happening in the metaphase. Write the significance of mitosis. It is also based on the marks allotted. You can write 1 or 2 or more than 2, 3 and all. Okay, it is based on the thing asked, based on the marks asked. With that, I am coming to an end. Hope you understand this chapter properly. If you want any clarification, do visit our YouTube channel, and we do have many other videos also. So, if you have any doubt, please re-watch this video again. With that note, I am taking a leave. Thank you very much. Om Shri Sai Ram.